Hello and welcome to this CosmosID webinar on microbiome R&D in skin health. My name is Arne Materna, I'm VP of Product at CosmosID, and I'm looking forward to exploring with you over the course of this webinar the microbiome R&D workflow in the context of skin health and skincare product development. We would like to invite you to submit your questions using the chat function. Our team will field your questions and compose answers, and since we would like you to be able to revisit them at a later point in time, you will receive the answers via email. So let's get started and look into the topics that we're going to cover as part of this webinar. After an introduction that'll cover the microbiome in the context of skin health and disease, we're going to dive deeper into the different segments of the microbiome R&D pipeline for skincare product development. Next, we'll explore a case study on the skin microbiome in psoriasis, and finally, we'll review microbiome R&D essentials. The emergence of next-generation sequencing, well over a decade ago at this point, had really a profound effect on microbiology and has really revolutionized our understanding of microbial ecology. We today know that the human host and virtually every other environment is teeming with often very diverse microbial communities. And while we historically considered microbes often in the context of pathogenicity, we know today that the human microbiome contributes vital health benefits, such as the regulation of our immune system, the biosynthesis of vitamins. It contributes a healthy lipid as well as carbohydrate metabolism, and can act as a barrier against the colonization with more dangerous pathogenic microbes. Conversely, a disruption of the healthy host microbiome homeostasis has been associated with a broad range of diseases. For example, neurological and neurodegenerative disorders, autoimmune and chronic inflammatory diseases, GI diseases, metabolic disorders, as well as oral and skin diseases. And as a consequence, the microbiome has emerged as a target for the development of novel diagnostic and therapeutic approaches. When considering the skin microbiome for developing new microbiome interventions, it is important to quickly look at the interplay of skin and microbiome. Both together, the skin and the microbiome together, contribute an important protective barrier function. This barrier function is blocking toxic environmental factors as well as invading pathogens. Our skin microbiome consists of bacteria, viruses, fungi, and mites, and the skin is colonized with these organisms at birth, but the microbiome composition is changing throughout our life and depends on our diet, the environment we live in, or our lifestyle. It is also important to note that the skin microbiome, which is affecting the skin barrier function, is interacting tightly with the immune system, which in turn can affect the skin barrier and other skin functions. Disrupting the skin microbiome, also referred to as dysbiosis, has been associated with unhealthy changes in diet, lifestyle, or environment, with, for example, hormonal disbalances, or the use or overuse of certain products. And possible consequences associated with dysbiosis have been acne, atopic dermatitis, psoriasis, and many other conditions. A range of different interventions have been or are being developed, which are shown here on the right-hand side of the slide. They can be grouped into different categories. The first category shown here are prebiotics. Prebiotics include ingredients that induce bacterial growth. Probiotics, in contrast, include microorganisms, typically living bacteria, that are believed to provide certain health benefits. Postbiotics are bacteria-derived ingredients, such as enzymes, peptides, or lactic acids produced by microbes associated with the skin microbiome that are believed to confer these benefits. Another label that you see increasingly often is the label biome-friendly. This label is associated with products that do not claim to confer certain health benefits, but that have been tested to not disrupt a healthy skin microbiome. And finally, there are traditional pharmacological approaches which fall into the realm of therapeutics. Now, let's move on and explore next the different stages of the skincare product development process. Historically, the skincare development process started with a target discovery to really identify your ingredients, which then become part of your formulation. Once your formulation is locked down, you have to demonstrate safety to obtain regulatory approval, And ultimately, you have to consider, how do I maintain a high degree of quality as I'm scaling up my production. However, there's an important trend in the consumer product market, and that trend is a trend towards an increasing demand for scientific evidence. Consumers today are microbiome aware through the news. They conduct in activities such as personal health tracking. And not only healthcare is increasingly personalized, but also personal care. And as a consequence, clients demand more and more often clinical evidence for the effects and the safety of the products that they use on a daily basis. Historically, the brand and the perception of your brand in the mind of the consumer was what really dominated the effectiveness with which you penetrate a market with a new product. And the lower left-hand quadrant here, where you have poor brand equity and no scientific evidence, always has been a non-starter. However, driven by developments in nutrition and nutraceuticals, skincare companies have emerged that, as they're building their brands, providing hard scientific evidence for the value of the products that they are developing. And as a consequence, larger companies with well-established brands are now struggling to keep up, providing also scientific evidence for the products, especially the novel products they're developing. And as a consequence, this traditional pipeline is becoming more complex. Specifically, target discovery as a starting point is no longer enough. We now have to also consider the function of the targets or the ingredients that we're selecting. And it is no longer sufficient to demonstrate safety to the regulators. We also need to demonstrate efficacy of the targets or ingredients that we've identified, at the very least to satisfy the demand for scientific evidence by the consumers. So it is important to identify what it takes to be ready to improve skin health through microbiome interventions. Over the next couple of slides, we're therefore taking a detailed look into how microbiome technologies can contribute to the R&D journey. We'll start with the technologies involved in target discovery and then move to the technologies involved into identifying target function. From there on out, we move into safety, demonstration, the demonstration of efficacy, and the QC steps necessary for scaling up production. But let's begin with target discovery and the microbiome profiling stages that play an important role during that phase. We often emphasize, and it has been in the recent years commonly accepted, that the clinically informative and actionable unit in microbiology is not a genus, and it is not a species. Ultimately, the ingredient that you're using or the target that you're trying to affect is a strain. Here are some examples that hopefully exemplify this statement a little bit more. Consider the species of Lactobacillus casei, which is a species that is widely used in food production, where one strain is involved in the fermentation of cheddar cheese, another strain in the production of lovely Chardonnay, and yet a third strain in the production of a breakfast drink. And while you may try to argue that if you get your strain wrong, the outcome is at least still, delicious all around, that is very much not the case if you consider another species, the species of Escherichia coli, where certain strains convey important functions and are part of our commensal gut microflora. They're involved in vitamin biosynthesis and provide colonization barrier effects against overgrowth of otherwise dangerous pathogens. However, other strains, such as O157H7, are very, very dangerous and cause hemorrhagic enteritis, which is a potentially lethal disease. So as you can see, the difference between being able to discriminate at strain level and not can be the difference between life and death. And in order to provide the ability to deliver strain level resolution, CosmosID has invested heavily in an analytical platform. We have invested over 10 years of curation into what we believe to be the world's largest and most comprehensive database that involves over 150,000 entries and does not only cover pathogens, but importantly also commensals as well as environmental microbes. And what sets this database apart from other databases that are out there and are available is the structure and the approach to curation that we take. We do not only include organisms that we find in public repositories and associate them to the assigned taxonomic names that are available. Instead, what we do is we reinfer the phylogenetic relatedness of all the entries in our database to establish effectively a phylogenetic tree of microbial life. That is where a lot of the IP and computational heavy lifting on our end resides. And this tree tells us two important things. It tells us, for once, where do we have microbial lineages that are maybe underrepresented? And this is where we can engage in de novo sequencing projects or collaborations with biobanks to backfill our databases. The second thing that it tells us is possibly even more important, and that is it gives us a hierarchy or topology. And this structure or this hierarchy is the hierarchy we use when we define the ontology or structure of our database. And that allows us to do something very important. It allows us to traverse all the entries in our database and look for biomarkers, in our case variable length k-mers, that are uniquely identifying organisms at each level in this phylogenetic tree, meaning we have biomarkers that uniquely identify organisms at all the way down at strain level. And we have biomarkers that are shared among strains in the same species or subspecies, but that are again unique at the species or subspecies level, and so on. And so that allows us to very sensitively detect organisms and characterize them at strain level and confirm the call by looking for shared biomarkers upwards the database structure. And as a consequence, we have very high specificity and strain level resolution without sacrificing sensitivity. This approach has been applied to curate databases not just for bacteria, but also viruses, fungi, protists, a range of specialty databases, as well as databases for antibiotic resistance genes, virulence factors, plasmids, and phages. We frequently update the databases, and then when we do so, rely on published best practices for benchmarking to ensure that the database updates contribute to the high performance of our platform. When you're looking at this chart, this shows the results of one of those benchmarking expeditions. You see here in light blue the values for precision, in light green the values for sensitivity, here labeled recall in the legend. And what becomes apparent as you're looking at the upper chart on the left-hand side is you can see that as many of the public algorithms are seeking to optimize either precision or recall, they do that at the expense of the respective other value, which is either lower in its average or more variable. And it is therefore important to not just look at sensitivity and precision, but also to look at the harmonic mean of those two values, which is called the F1 score. The F1 score really shows you how well are you doing in terms of precision and sensitivity, and how big is the trade-off that you're making between those two as you're trying to optimize your algorithm. So in a nutshell, the higher the F1 score, the better your performance at sensitivity and precision, and the lower the trade-offs that you're making. And what you can see at species level is that CosmosID is performing very favorably. However, as you're moving to subspecies and strain level, this is really where the platform begins to shine and everything else starts to fall off very drastically. Now, we do understand that in-house benchmarking using your own methods and known data sets makes you vulnerable to confirmation bias, which is why we've embraced in the last 2 years third-party controlled blinded validations. And we've submitted several times our pipelines to such efforts. And what you see on that slide are the 3 most recent efforts that we've participated in. Those validations were carried out by Janssen Pharmaceuticals and by 2 different centers of the US Food and Drug Administration. And we're very happy being able to report that we won all 3 challenges with leading scores in strain level identification. Now, good analytics is important, and good analytics can be used and has been used to provide scientific evidence for skincare products. I want to point out that a metric that has been reported frequently is alpha diversity. Alpha diversity is a measure of species richness, and scientific evidence for skincare products is currently focusing a lot on whether the products contribute to species richness in skin that has been affected, or at the very least doesn't disrupt and reduce species richness in biome-friendly products. However, for developing microbiome-based skin health interventions, alpha diversity is not enough. It is important to also look into microbiome composition and to understand how does the composition change at strain level between diseased, healthy, or the course of an intervention. And to obtain this information, a process called microbiome profiling is used. Microbiome profiling uses as input the raw sequence data and generates data sets that tell you which microorganisms or which microbial functions are present and at what abundances. And having accurate microbiome profiles that are resolved ideally to strain level resolution then allows you to engage in the second step, which is comparative analysis. Again, I'd like to stress that the accuracy of the data that go into comparative analysis is very important, as it determines the validity of the findings that you make throughout that process. The first step in comparative analysis often is beta diversity or between species diversity analysis. To do that, we use a process called principal coordinate analysis. And what principal coordinate analysis seeks to do, you see that chart on the lower left-hand side of the slide, is it seeks to separate all the samples maximally based on their compositional dissimilarities. So this process draws a two-dimensional or three-dimensional space drawn by eigenvectors that really maximize the distance between samples based on compositional differences. And as a result, if you see that samples belonging into the same cohort, for example, healthy, intervention, diseased, cluster together, this is evidence for compositional differences occurring consistently between those cohorts. And that is then the trigger for the second step, which is differential abundance analysis, to determine which organisms or which functions most contribute to these observed differences, and to explain also which of these functions are acquired over time or stable over time. Lastly, we move into predictive analysis as the third step, which uses differential abundance data as input. However, then it goes a step further. It considers two things. It considers the statistical significance of the differential abundances obtained for those important organisms or functions, and it considers the consistency of these differences being in line with the biological hypotheses. And as a result, it reports taxa or functions that best explain the differences between your cohorts, such as healthy, intervention, diseased, and so on. Now, once you've identified your targets, the next important phase is to understand and delineate the mode of action by looking into the function that the targets can contribute to the human host. Traditionally, DNA sequencing is used to understand the human microbiome. DNA sequencing begins with obtaining a biological sample of the human microbiome, which is then converted molecular biologically into a sequencing library. And the process of DNA sequencing converts that biosample into a digital sample, which can then be interrogated through bioinformatics. Depending on the sequencing method and the bioinformatic method that you use, you can not only obtain strain level or taxonomic abundance profiles, but you can also obtain abundance profiles of functional genes observed in the microbiome as an approximation of function. Now, what we see, not yet in the skincare space, but in the pharmaceutical and nutritional space, are efforts that are seeking to obtain independent lines of evidence for function, to move from prediction really to observation. And in addition to sequencing taxonomic profiles, these groups are starting to also consistently generate functional microbiome profiles and back them up with, for example, metabolic profiles that are obtained, for example, through mass spectrometry. And what you then can do is you can map those independently obtained data types, such as microbiome function, taxonomic microbiome composition, and changes in metabolites, to directional relationship networks, such as metabolic pathway maps, which allows you to generate hypotheses in a much better fashion than by using any single line of evidence by itself. So the microbiome function provides insight into the mechanisms or consequences of disease and delivers a mode of action of the intervention that you are developing. Now, let's move on to the third step, which is the demonstration of safety. Since you're using microbial ingredients, metagenomic sequencing is important and can help assess whether your ingredients contain, in addition to the strains of interest, also known pathogens, or whether certain genetic elements, such as antimicrobial resistance or virulence that can be transmitted between organisms, are present or have emerged in your ingredients. Another important aspect, in addition to safety, is demonstrating the efficacy of your product. And I'm going to show you an example of a project that we've carried out with a client of ours, Synthetic Biologics, which is a company that is developing a microbiome protection drug that is protecting, in this case, the gut microbiota from the hugely disruptive effects of antibiotic use. What you see in this chart are essentially microbiome profiles, and you see 3 different experimental groups or cohorts. On the left-hand side, you see the group that was the control group, and you see a healthy microbiome composition. In the middle, you see the group that was treated with ceftriaxone, in this case, an IV antibiotic. And on the right-hand side, you see the group that was treated with the antibiotic plus the experimental microbiome protection drug candidate. And the results were rather striking. We were able to measure not only that the disruption was reduced compared to antibiotic treatment alone, but what we also could show was that the drug was preventing the overgrowth of, in this case, Methanobrevibacter smithii, which at low levels is a gut commensal, but as it causes overgrowth, it contributes to secondary infections, and the drug was able to ameliorate and reduce that impact. And here you see the reduced disruption of the healthy gut microbiota. Finally, once you've demonstrated safety and efficacy of your candidate, you're going to be challenged with aspects of maintaining a high level of quality during your production process. Again, metagenomic sequencing can help, and this is an example where we applied metagenomic sequencing to establish, for example, that ingredients and labeling claims are correct. This is an example of 4 different probiotics. On the left column, you see all the strains that were claimed by various labels to be part of the ingredients or the formulation. And in blue, you see which organisms should have been contained by each antibiotic per label. And in fact, metagenomic sequencing is able to confirm these claims with extremely little noise. Continued safety is another important aspect where metagenomics allows you to routinely survey whether contaminations have emerged in your formulation. Those could be bacterial or microbial contaminations, contaminations of emerging antimicrobial resistance or virulence, all of which could pose a health and safety threat to your consumers. Now let's move into the case study that explores the microbiome in the context of psoriasis. What we did is we took a very well-received publication that came out of the lab of Nicolas Segara that is exploring diversity and compositional structure of the skin microbiome associated with psoriasis. The authors were able to collect both affected and psoriatic skin from 4 different body sites, the left ear, the right ear, as well as the left and the right elbow. And the method that we've used to reanalyze the author's data is essentially the CosmosID bioinformatics pipeline, which is available to you under app.cosmosid.com. You can sign up for a free account, explore the software, or contact us for a demo. We're very happy to provide. Now let's look into the data. At first, we're able to confirm the author's findings in that sense that alpha diversity in healthy versus affected skins was changed. The alpha diversity in psoriatic samples on both body sites was significantly reduced compared to unaffected skins. So there was a collapse in species richness. The authors also reported that psoriatic samples presented an increase in the relative abundance of Staphylococcus at genus level. And that is also what's shown here, where we're comparing the healthy cohort to the diseased cohort at the bottom, and you see an increase in relative abundance of the genus Staphylococcus that is consistent between cohorts and across body sites. Now, as we're comparing the compositional differences between the different cohorts using beta diversity analysis, we see very little clustering. In this case, we're trying to resolve 4 different cohorts. Here we're collapsing 2 body sites that are healthy and 2 body sites that are affected. And again, we see very, very little clustering, which necessitates a higher resolution into investigation to identify which species or maybe strain level differences are actually contributing to the psoriatic microbiome. As I mentioned initially, it is important being able to not only resolve microbiomes to genus or species level, but to strain level for that very reason. So we're diving on the next couple of slides into an increased resolution analysis. Here we're looking into the genus Staphylococcus at greater resolution. And what we find is not just an increased abundance of the genus, but we see that at species level, the diversity of the genus Staphylococcus is collapsed and reduced and may play a role in the psoriatic microbiome. This chart depicts the differences between diseased in red and unaffected in green, and it shows that the Staphylococcus species, including Captitis and aureus, show increases in relative abundance by on average 7 to 8 percentage points. However, not all Staphylococcus species are in fact increased. Some species, notably Staphylococcus epidermidis, is severely reduced, in fact by over 20% in relative abundance, showing that you have to dive deeper in order to understand how changes in composition may contribute to the disease. In addition to Staphylococcus, the authors also noted variations in Cutibacterium acnes and Propionibacteria between healthy and affected body sites. Here you see an overabundance of Propionibacteria and a reduced abundance of Cutibacterium acnes. Let's move back to that chart that explores species-level abundances, and we were able to once again confirm that relative abundance for Propionibacteria as well as for Micrococcus luteus at the species level were increased by about 10 percentage points, while for Cutibacterium acnes we observed a severe decrease of 12 to 13% in relative abundance. Now we're shifting our resolution once more, and we're diving from species level down to strain level. And what we're finding at this level is rather interesting. For Staphylococcus species, we already had mentioned that we saw increases for Captis and aureus between 7 to 8 percentage points, and we're reporting here strains with elevated relative abundance, which include M1382, QN1, and CR01 for Staphylococcus captitis. For Staph epidermidis, which was severely reduced at species level by over 20%, strains that contribute to that reduction include AU1203, VCU071, SCL25, and SCL31. However, not all strains contribute to that severe reduction in abundance. There's also certain strains with distinctly elevated relative abundances, including NIH LM049, BCM HMP 0060, and NIHL M039. That is important because strain level resolution in this case enables you to tell differences between strains even within the species level that may contribute to the disease or the disease progression. Let's shift our focus to Propionibacteria and Micrococcus luteus, which both were increased at species level by 10%, reporting here again the strains with elevated relative abundances, some of which have not been classified. And as we're looking into Cutibacterium, some of them have been reclassified as Propionibacterium acnes. We saw a severe decrease at species level by 12 to 13%, as you remember. And the observed strains that contribute to that decrease are listed here. However, once again, similar to Staphylococcus epidermidis, not all strains contributed to that reduction. Some strains were significantly elevated in their relative abundance, including Propionibacterium acnes HL020PA1 and an unidentified Propionibacterium. The authors had run into a similar issue and were able to list in some cases only purely characterized Propionibacter acnes or Cutibacterium acnes species that couldn't be further classified. That is why we augment our typical classification with a secondary classification step. Our typical classification uses a highly validated database of organisms for which a lot of evidence, both from a genomic perspective as well as a lab culturing perspective, exists. However, recently a novel approach to identifying new microbial taxa has been proposed that relies not on lab methods, not on culturing methods, but solely on genomic methods. These so-called metagenomic assembly-derived microorganisms or microbial genomes come from environmental samples that have been sequenced and subsequently bioinformatically analyzed to obtain hypothetical genomes from these environmental data using de novo assembly processes. So we've developed a dark matter database as a secondary lookup against organisms that we can't classify by conventional means, and the results are shown here on the right-hand side. What you see is we're able to further classify 3 of the Cutibacterium acne strains down to species level. And what is confirming or supporting this classification outcome is that if one strain was present in any given sample, the others were not. So we didn't have any redundant calls. However, as you can see, if you group them by cohort, none of the 3 Cutibacterium acne strains was disease relevant in this data set. The authors also identified the fungal genus Malassezia. Using the CosmosID fungal databases, we were able to identify Malassezia as well and resolve it to species and strain level. And we're listing here the most commonly identified species and strains. And as you can see down here once again, if you're sorting by cohort, is that in this case, none of the 3 strains were disease relevant. So in summary, alpha diversity of skin microbiota from affected samples is significantly reduced, which both the authors and us were able to show. A lack of significant beta diversity clustering, however, necessitates to look for individual taxa with differential abundances at better resolution. The species trends that were observed by the author were also confirmed here, and we were able to further resolve them to strain level. And strain level analysis ultimately is necessary to understand the full picture in psoriasis, especially for Staphylococcus epidermidis and P. acnes, where we were able to show that the overall trend at species level requires further resolution, as some strains are supporting the trend, however other strains are moving into the other direction. The CosmosID K-mer-based analytical approach and our databases can classify not just cultured and well-characterized, but also additional fungal and bacterial species that were missed in the original paper, even those occurring at low abundances. The dark matter databases can contribute to the classification of previously undescribed strains. And one thing I'd like to point out, which we haven't shown here, is that the platform not only reports microbiome profiles with strain level resolution, but it also reports virulence factors as well as antibiotic resistance markers, which may be relevant in the context of this disease. So with that, let's switch to the last part and recapitulate different R&D essentials required for the development of microbiome-based skin health interventions. We believe it is very important, if you carry out microbiome analysis, to control the entire end-to-end workflow, beginning with sample collection, preservation, storage, DNA extraction, library preparation, sequencing, and analysis, as all these steps are interdependent and can affect the outcome at the end. To that end, we've established a CLIA and GLP certified facility that is developing, validating, and optimizing methods for shotgun, amplicon, and isolate sequencing. And we have augmented these lab capabilities through the development for many years and the curation of what today is the industry-leading platform for strain-level microbiome analysis. Challenges we can help overcome through the setup is to offer a standardized and controlled sample collection, processing, sequencing, and analysis of your microbiome data. We'll resolve microbiome data to strain level. We can carry out functional analysis and resistance profiling, and if you'd like to move in that direction, support you with metabolomic analysis. And we provide you with analytics that we do not only keep in-house, but move to the client side through a software portal, which you can find under app.cosmosid.com, which provides the best-in-class analytical engine for microbiome R&D. The ultimate goal is to help you drive actionable results and discoveries in a way, however, that is controlled and reproducible. So in summary, what is needed to improve how we generate high-quality insights, testable hypotheses, which are key to microbiome product development, is to really control and support the full process from study design, sample collection, sample preparation, sequencing, all the way to bioinformatics and the interpretation, for instance, using machine learning based methods to support you end-to-end from sample all the way to data analysis. With that, I'd like to invite you to submit your final questions using the chat function, or if you've run out of time, feel free to send us your questions later via email to info@cosmosid.com. And with that, I'd like to thank you for joining today's webinar. Thank you for sending your questions and your attention. I just hear that we have time to answer some questions now, so I'm obviously happy to do that. Here comes the first question from my colleague. What is CosmosID's position on swabbing versus tape stripping? That's a good question. And as you know, in the sampling of skin specimen, swabbing and tape stripping are two methods to obtain the skin microbiota of the skin. And tape stripping is a more invasive approach that is to some extent harmful compared to swabbing. There is several studies that we're aware of that are forthcoming that suggest that there is no statistically significant difference between the two approaches, but we're obviously monitoring progress in that area closely, and we're experienced with both technologies. Next question. How deep should one sequence skin samples? That is also a very good question. As you know, sampling skin comes with sampling of skin cells that contain human DNA, not only microbes which contain microbial DNA, which is the DNA that we're actually after. So as a result, we're dealing with so-called background DNA, quite substantial amounts. And so in order to sensitively detect microbes on the skin, it is important to sequence deeper than for other specimen types, meaning generating more sequence reads to capture all the DNA coming from microbes. We typically recommend to sequence, depending on the questions asked and the applications, between 20 million reads or deeper, up to 40 million reads. Do you have samples that are healthy skin microbiomes to compare other skin microbiome samples to? The answer is yes. We have access to skin microbiome samples from studies that are healthy and that we can use. However, it is always important to use a baseline that comes from unaffected skin, ideally in the same study subject, as the environment obviously affects the baseline microbiome as well. And if you look for differences that are disease specific, the best practice is to obtain samples from affected and unaffected skin of the same individual. Next question. How do you deal with all the host contamination from swabbing? Yes, that's a very good question as well. So host contamination is, as I said, from skin swabbing is unavoidable. The way we're dealing with this is typically by using a special enzymatic cocktail that increases the lysis efficiency of bacterial cells. Bacterial cells can be harder to lyse than eukaryotic cells, meaning the DNA yield you obtain from bacterial cells can be lower. So by making an extra effort doing DNA extraction using special enzyme blends, maximizing the yield of the bacterial DNA can reduce somewhat the overcrowding in the DNA sample of the human DNA. So that is one way we can maximize sensitivity and keep the impact from human background DNA to a minimum. Now, for diagnostic applications where sensitivity and the lower limit of detection are important, there is the possibility to carry out host DNA depletion experimentally. However, using this for microbiome analysis and microbiome studies is not advisable, as it will affect the composition of the skin microbiome samples. So for diagnostic applications where you try to sensitively detect a certain organism or group of organisms, host depletion is an option. For microbiome studies where you're seeking to understand the differences in composition, we advise against that option. Any other questions? How fast do you process samples? Our lab is set up to turn around samples for non-diagnostic applications in 3 to 5 business days. However, our typical turnaround time is around 4 to 6 weeks. We can deliver samples faster. However, at that point, we have to charge more. Are you operational during the COVID outbreak? Yes, that is another very valid question. Since the CosmosID laboratories in parts offer diagnostic services, so one part of our lab is a reference laboratory, the other part is part of the contract research organization. We have the safety standards to deal with infectious samples, and given that we offer diagnostic services, it is very important that we maintain fully operational. So we're deemed an essential lab. We're fully operational. We're dealing with the outbreak by reducing presence of our employees at any one time, spreading out the shifts as well as the days over longer periods to increase the social distancing and provide a maximum of safety, in addition to some other precautions that we've taken. But yes, the lab is fully operational during the COVID outbreak. Maybe a last one. What sort of claims can we make from doing a microbiome study? That's a very good question. The claims that are commonly made in the skincare industry, as I mentioned earlier, are claims with regards to microbiome friendliness. This is where we're looking for the disruptive effect of skincare products on the healthy skin microbiota. Claims that there's also health claims. However, there you're entering regulatory or regulated space, which, however, can also be made for therapeutic or medicinal skincare products, where you're not only proving a safety, but you're also providing evidence through clinical trials for the efficacy of your formulation. This is something that we regularly do, but typically more often at the moment still in the pharmaceutical industry, but we see these kind of requests more commonly in the skincare industry. And okay, maybe the very last one, why should we use shotgun instead of 16S? It's a very good question. 16S and shotgun are two very different methods to explore the microbiome. 16S only looks at one gene, shotgun looks at the entire genome. So with shotgun, you can obtain much more information. You can identify organisms with strain level resolution, so you know exactly which organisms are there. Whereas with 16S, you're looking at one gene, and so you can roughly estimate what kind of organism it is. We use the term species to genus level resolution for 16S compared to strain level resolution for shotgun. Shotgun also allows you later to obtain information on the functional aspects of the microbiome, so which metabolic capabilities are encoded in the genes of the microbiota. This is information that 16S cannot really derive well from the samples, as it looks only at one gene. However, 16S has the advantage that it requires very, very low DNA input amounts, and sometimes microbiome sampling from skin yields only low amounts of DNA. So 16S can be the saving grace. And it is a method that's perfectly suitable if you're not looking to resolve organisms at strain level, but maybe just try to understand how does alpha diversity or species richness change at species to genus level. So if you're dealing with low input amounts, 16S is an option, but it can't give you all the answers. But it could be a first step towards certifying microbiome safety, for example. And with that, I'd like to thank you for attending, for all your questions. Let us know if you have any further questions. You can see here our contact information. We're looking forward to your messages if you'd like to get in touch with us. Thank you very much.